Hello, hello, hello.
Can I start? Voice is okay. No, it's start. Hello, students. Today is our very first chapter in class 12th, reproduction. In this chapter, we have to read three topics. First one is sexual reproduction in flowering plants. Second is our human reproduction, and third one is reproductive health. Right. So before start with this unit, some information is given here. What is the information? Simply we read this because every line is important in NCERT. Biology is the essence in the story of life on earth. What is biology in a term, in a statement is given biology in essence. It is the story of life on earth, right? While individual organisms die without fail, it is a, we know inevitably it is a correct that everyone who come on the earth, he should be die. Ultimately, he die. Either it is a plant or it is an animal. Species continue to live. That's why reproduction occurs. Because species continue to live. Organisms, it die. Then a new organism form. New organism form is what? How it continue its species. How it continue its species. Right? In future, it is through reproduction. Right? To live through millions of years unless threatened by the natural or anthropogenic in extinction. Look here, one word is coming here, anthropogenic, right? Anthropogenic means what? Anthropogenic word means man-made, right? Because this time what happens? We use pesticides. Through pesticides, we kill the insects. Insects means which are harmful to us, which are harmful to our plants or crops, right? When we that means destroy them, what happens? New offspring will come, right? How they new offspring come? Because they reproduce through sexual reproduction. Look here, anthropogenic means man-made, right? Extinctions. Reproduction becomes a vital process without which species cannot survive for a long. Species, not a organism. Species cannot survive for a long. Means without reproduction, either it is a asexual vegetative or it is a sexual. If no reproduction means no multiplication means what happens? After one generation, means it is totally extinct, right? Its progeny by asexually. Sexual means, right? They forms. Sexual mode of reproduction enable creation of a new variants. In sexual reproduction, what happens? Fusion of male and female gamete, and these female and female gamete, they come from the different organisms. That means what? In different organisms, we have different variations are there. Means they have different characteristics are there. So when they intermingle with us, what happens? In new generation, we found some variation. Means some dissimilarity will also come, or we can say a intermediate organism is produced, right? And this leads to variation, right? So that survival advantage is enhanced. Variation come means it, we can say also there is a adaptation. It is useful or it is harmful, both type it may be, right? But what happens if it is useful? Useful means it is adaptable according to our environment. Then what happens? These organisms can survive easily, right? The unit explained in this unit what we read here. It explains reproductive process in flowering plants. Flowering plants, you know, here one word is coming, flowering plants. So what it mean? Flowering plant means which plant? They are not a lower plant, means which have flower. Flower means angiosperm. Angiosperms, they are called as flowering plants, right? Okay, why I am not saying about a gymnosperm? Gymnosperm is also a, we cannot say they are not flowering plant, but what happens in gymnosperm? Sporophylls are there, right? And these sporophylls means they bears sporangia. Okay, these sporophylls, they form a cone-like structure, or we can say strobilus. So in gymnosperm, cone or strobilus is formed, right, which is different from flower. But in angiosperm, It is typical flower is present only in angiosperm. That's why here we are going to read about the angiosperms that is division, right? Flowering plant and human. Human, we also. As easy to relate the representative examples. With examples, a related perspectives on human reproductive health also, because it is also important for human. We are human. We have to know about the reproductive health and how reproductive ill health can be avoided in is also presented to complete our understanding of biology of reproduction. Means biology of reproduction not only this one reproduction, but also the evil or we can say ill health of reproduction, right? Means what is reproductive health? It is also very important for us because a new offspring will come through this reproduction. Okay. So we start our first chapter, that is our sexual reproduction in flowering plants. The scientist given here is Indian Panchanan Maheshwari, right? Why he is very important here? Panchanan Maheshwari belongs to Jaipur, Rajasthan, right? He is the person who bring out the very first textbook of biology. Textbook for biology means for secondary schools, right? Which one? For higher secondary schools, published by NCERT in 1964. After Kothari Commission, right? So Kothari Commission, he is the leader means for bringing out very textbooks of biology for higher secondary schools published by, right? And he also a writer of embryology of angiosperm, right? He is the writer of embryology of angiospermic plants. Panchanan Maheshwari. He is also considered as father of Indian embryology. Now, sexual reproduction in flowering plants. In this chapter, we have 5 aspects which we have to read. That is flower, a fascinating organ, right? Second, pre-fertilization, because we know here when we consider the sexual reproduction, we have 3 phases. 3 phases means what? First phase is our pre-fertilization stage, then after fertilization stage, and then post-fertilization. In pre-fertilization stage, what happens? Formation of gametes. Here 2 processes there. Formation of gametes, because it is a plant, means microsporogensis and megasporogensis. Second, transportation of gametes, that is pollination and germination of pollen tube. Third is then after, okay, these 2 process happens in our pre-fertilization stage. In fertilization stage, what happens? Fusion of gametes. And the third stage, it is called post-fertilization stage. In post-fertilization stage, formation of zygote, and then embryology. The embryology is development of a seed, and after then germination, right? So here, pre-fertilization structure and events. 2 events. Which events? In this portion, we read about the microsporogensis and megasporogensis, means formation of gametes, and transportation. Transportation means what? Pollination and germination of pollen tube also. Here, double fertilization, right? That is syngamy here, because it is an angiosperm, so triple fusion and double fertilization, right? In this post-fertilization structures and events means formation of embryo and development of embryo. We can say development of embryo and seeds or fruits development. Apomixis and polyembryony, it is another term. Apo means what? Without. Mixis means without fusion, without fertilization. We can say without fusion. Most important, without. Formation of seed without fusion, that is called apomixis, right? Apo means without, amix means fusion. It is actually it is a that means a critic about against the sexual reproduction, right? And polyembryony, poly means many, embryony means in a single seed more than one embryo is formed. That is called polyembryony. As for example, in our citrus means lemon or orange. Okay, we will read this one by one. First, what is given? Flower, a fascinating organ of angiosperm. In this passage, nothing is important here, but we have to read only one. Are we not lucky that plant reproduce sexually? Yes, we are lucky, because because sexually means what? We have many variations are there also. The myriads, and they are sexually means we have flower, right? We get flower. Myriads, flower, we get fruit, seed. These are all the product of sexual reproduction. Seed, fruit, flowers, and all these are very important to us, because food which we get, if it is a grain, that means it is our seeds. If we get fruits, fruits means vegetables means what? They are fruits. We get our food in the form of fruits or seeds, and these are the reproductive products, right? Flowers we also use here. The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we soon over, the rich colors that attract us, we attract in flower when we go garden, right, are all there as an aid to sexual reproduction. So flower do not exist only for us to be used for own selfishness, but all flowering, right? All flowering plants show sexual reproduction. A look at the diversity of all flowering plant means what? All angiosperms, they shows sexual reproduction. A look at the diversity of structure and the inflorescences, flowers and floral parts shows an amazing range of adaptations according to the environment to ensure formation of the end product of sexual reproduction. End product means what? What is given here? The fruit and the seed. End product means what? The fruit and the seed. These are the end product, right? In this chapter, let us understand the morphology, that is external structure maybe internal, and the process of sexual reproduction in flowering plants. Flowering plant means what? Here it is given especially angiosperms, right? So we start here from our first point, that is flower, a fascinating organ of angiosperm. Human being had, what is given? Human being have had an intimate relationship with the flower since time immemorial. We cannot understand it, at least we have no idea from how many years because flower it is attractive, it is not attractive for humans only, animals also attract with this flower color. It memorial time is also. Flowers are object of aesthetic, ornamental, social, religious, cultural value, right? These are, we use flower in aesthetic, ornamental, social, religious and cultural programs, right? They have always been used as symbol of conveying important human feelings, right? We give flower to whom? For our loved ones, to our friends, or friendships, right? Such as love, affections, happiness, grief, mourning. When a person dies, that time also we use flower, right? From birth to death, and from our emotions, they are, that is the color of the flower, and the type of the flower, and the flower, they are totally means what? They shows our emotions also. That is given here, right? Now here given list at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. You can understand 5 flower names, you can write this one, right? I did not think that I have to tell you. Find out the names of the 5 more flowers that are used in social and cultural celebration in your family. Have you heard of floriculture? That is important. What is floriculture? Floriculture, right? What does it refer to? What do you mean by floriculture? Floriculture is a part of agriculture, we can say. Floriculture means flori means what? Flower, and culture means to cultivate the flower. What is floriculture? Floriculture means to cultivate the flower. Cultivate the flowers. Because this time the demand of flower is increased, because in industrial areas also many scents, or we can say sticks, right? That is in Hindi we can say agarbattis, right? These are many perfumes, some creams. They are also that is rose water. So different type of products are formed, industrial area also, and aesthetic manners is also in berry temples or other things. That means what I am only saying, this time demand of flower is increased. That means this floriculture is also increased in demands, right? It's a farmer, now he moves toward the floriculture, right? To a biologist, flowers are morphological and embryological marvels. Morphological means structurally, external structurally, and embryological means what? How they develop, means how a embryo is formed, how a new organism is formed, right? Because we get variations. How we get variations? From hybridization, and hybridization it is only possible through flowers, right? Marvel and the site of sexual reproduction. Flower is also called as the unit of sexual reproduction, right? In earlier classes, you have read the various part of the flower. Figure is given here, right? Now here, can you name the two parts in a flower in which the two most important units of sexual reproduction develop? So these two parts of flower, what are they? One is our Androecium and another one is our Gynoecium. Androecium and Gynoecium. Commonly, Androecium is called as stamen and Gynoecium it is also called as pistil, right? These are most important sexual reproductive plants. Other means what? Calyx and corolla, or we can say petal and sepal. They are the accessory part of the flower, right? A figure is given. Now look here, this figure. Right? What you see in this figure? You can see these are filaments and this is anther. Right? Filament and anther, they forms stamen. Means we can say stamen has two parts. One is anther and the other one is filament. Right? Second here, what given? This is colorful parts, means petals. Androecium is member of Androecium is called petals, because in morphology we already read this one. These are sepals, right? Sepals, it is the member of which one? Calyx. And this is petals, means it is a member of corolla, right? And this is a member of calyx, right? These anther or stamens, filaments, they are the member of Androecium. And this middle part, okay, this middle part, right? This one is stigma. This is our style, and here it is ovary. And these three parts, they forms Gynoecium, right? Gynoecium. Stigma, topmost part, which provide platform for anthers, right? Second, style is there, and then ovary. Where in ovary, what happens is ovules are there. This is locules, ovarian locules, right? We also said them carpels. Okay, locules. In locules, what is there? Seeds are present, right? That part, this part is called thalamus. Thalamus, right? And this twig-like structure, structure is called pedicel, right? So these are the part of flower. Okay, I draw a figure and then explain you. This is a flower. This one is a flower and it's a different part, right? Look here at this side. We start from this one is pedicel, this one is thalamus, right? This is our ovary, this is locule. This one is ovule. Ovule, right? Ovule. This is style, stigma, right? So this is the part of pistil, or we can say gynoecium. Gynoecium, right? Now here, this one is our anther. This is our filament, right? These two, they forms stamen, or we can say Androecium, right? This is colorful part, that is single member. Because it is a single member, it is called as petal, right? And it is a part of corolla, right? Here look, this one, this is our part of calyx. Means here single member means what happens, it is called sepals. Single member means sepal, and it is a part of calyx. Calyx is the group, right? So these are the different part of flowers, which simply here, right, which we have to read. Okay, come back to the NCERT. I think you understand this one. So different part of flower. Then after, pre-fertilization or structure of events. Pre-fertilization means what? I already tell you. Pre-fertilization means what? We have two steps, two events are there. One is called as what? That is called because gametes are formed, so we said them gametogenesis. In gametogenesis, because we have two types of gametes, one is male gamete and another is female gamete. Male gamete is microspore and female gamete is megaspore. That's why here we said them microsporogensis and megasporogensis, right? Embryo, first here, embryo is the post-fertilization stage. Is gametogenesis, gametes are formed in before fertilization. So it is called gametogenesis. Gametogenesis is two types. If a microspore, means a pollen grain is developed, it is called microsporogensis, or we can say development of pollen grains or male gametophyte. Second is our macro, or we can say megasporogensis, right? That means formation of a embryo sac and egg cells, right? Okay. And second event is transportation of gametes. Gamete means which gamete is transported? It is male gamete, right? Pre-fertilization structure and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Decision is taken place by which? Flower or plant itself. Several hormones. These hormones are called as florigens, right? Several hormones and structural changes are initiated. Structural changes also, because here leaf is converted into different that is floral primordia, right? So leaf primordia is converted into floral primordia. That's why here several hormones. Hormones means which hormones? Okay, here I close this here, right? Several hormones means which hormones? It is floral primordia. That is several hormones. Means what? Florigens, right? And structural changes. Structural changes means what? Here leaf primordia is converted into leaf primordia, is converted into floral primordia. Primordium is what? Undifferentiated group of cells. They are called as primordia. Now, when leaf primordia is converted into floral primordia, in place of leaf, what is developed? It is flower is developed. Flower means outermost node, it forms a calyx. Second node, it forms corolla. Third node, it forms Androecium. And fourth node, it forms the gynoecium. Are initiated which lead to the differentiation and further development of floral primordium. That is I said, right? Inflorescences are formed which bears the floral buds and then flowers. Inflorescences are formed. Inflorescences may be cymodes or racemose is formed, right? If cymodes is formed, what happens? The apical part, the terminal part of stem, it is converted into a flower. And if racemose type of means lateral, that means we can say auxiliary types. That means lateral nodes, they develop into a flower, right? In cymodes, what happens? Flowers are laterally arranged, but in racemose, flowers are laterally arranged, but in cymodes, flowers, it is terminal part of the stem, it is converted into floral primordium and develop into a flower, right? In the flower, the male and the female reproductive structures, that is Androecium and gynoecium respectively, differentiate and develop, right? You would recollect and that the Androecium consists of a whorl of stamens. Androecium means normally it is called stamens representing the male reproductive organ, right? And the gynoecium, it represents the female reproductive organ. We know these are very easy points, right? Now here, first point is here, stamen, microsporogenum and pollen grain. Stamen, microsporogenum and pollen grain. What is the structure is given here? Figure 1.2a shows the two parts of typical stamen. We have two parts. This is a stamen and each stamen it consists of two parts. Two parts means one is our filament and another one is our, this is a structure given. One is filament and another one is our anther. This is filament and this one is anther, right? Look, one thing is there. When you see this one, because this filament, it is attached base at the base of this anther, right? Base. This one is called basifixed. We will discuss when I tell you about the anthers. That we have four types of fixation here. Means connecting or attachment of filament with anther. It is maybe adenate, basifixed, dorsifixed, or versatile. This one is anther because it has two lobes, one and it is the second. So it is called as bilobed, right? Because each lobe consists two theca means what? It is dithecous. And each theca consists two sporangia means how many sporangias are there? Because four sporangias are there. So it is called as tetrasporangiate, right? One more important thing, it is a filament, right? And when you see the filament, so this which part of this one and another this base, the base of the filament, it is attached with either epipetalous, it means petals on petals. If it is a epipetalous, if it is attached with petals, or it will be attached with thalamus, right? And the apical portion of this filament, it is attached with anther. Now here, which one is proximal and which one is distal end? This end is called it is called distal end, right? And this one is called proximal end, right? So now we can understand this is the distal end, means the distal end of the filament. We can also say distal end of the filament is attached with anther, and the proximal end of the filament, it is attached with either thalamus or if it is epipetalous by petals, right? Okay, come and read this one. What is given? Typical stamen, a long slender stalk called filament, and the terminal generally bilobe, right? Filament and the terminal generally bilobe structure called anther. Generally bilobe, mostly bilobe, but it is not always correct. Sometimes this single lobe is also there, right? Then it is called monolobe, unilobe, right? The proximal end of, but it is very rare, mostly bilobe structure here. Bilobe structure. Proximal end, look here. Proximal end, already I tell you, this side it is proximal. And this one is distal. What is given? The proximal end of the filament is attached to the thalamus and the petal of the flower. If it is a epipetalous flower, it is attached with petals, right? The number and the length of stamens are variable in the flower of different species, right? Number and length, because we know 5 stamens are present in which Solanaceae, right? 3 or multiple of 3 stamens are present in which Liliaceae or in monocots, Gramineae, right? 9 plus 1 stamens, they are present in our Fabaceae, Leguminaceae. 2 plus 4, they are present in our Cruciferae, right? And Cruciferae also, length is different, means tetradynamous condition is seen. In Salvia, not Salvinia, Salvia, we have didynamous condition is there, right? These things we already read in morphology of flowering plants. That is given here. Number and length of stamens are variable in the flower of different species. If we were to collect a stamen each from the flower from different species and arrange them on a slide, what we see, we will be able to appreciate the large variation in the size seen in nature. That's why what happens. That's why Androecium, that is Carolus Linnaeus, for his artificial classification, he used vegetative characters and also Androecium. This time also, because 9 plus 1, if 9 plus 1 that is died adelphus is present in Androecium, what happens? We understand this belongs to the Leguminaceae family, right? If 2 plus 4 is there, means tetradynamous condition is there, we simply understand this belongs to the Cruciferae family or Brassicaceae family, right? This is very important, the length and the number of Androecium. Carefully observation of each stamen under a dissecting microscope, making neat diagram would elucidate and the range in the shape and the attachment of anther in different flowers. When we see, that means when we cut a transverse section of anther, right, and place it into on the slide microscope, we can see the different layers of an anther, right? What is given here? Now here, in this figure, you can see this one is anther, right? This side it is left lobe, right lobe, right? Means what? 2 lobe are there. So it is called bilobe. Bilobe. First thing. Second, this one is 1 and 2. These are 2 theca. Look here, 1 theca and 2 theca. So how many thecas are there? 2 theca. Right? So it is called as dithecous. Dithecous, because 2 theca is there. How many sporines are there? These are sporanium in which pollen grains are stored or microspores are stored. 1, 2, 3, 4. Because 4 sporanium is there. 4 sporangium. We can say as sporangium. It is called tetrasporangiate. Do you understand? Tetrasporangiate, these 4 enzymes. Mostly angiosperms, they are angiosperm anther, they are bilobed, dithecous and tetrasporangiate. Right? But not all, because one exception is there, that is family Malvaceae. In family Malvaceae, what happens? Here it is bilobed, monothecous and bisporangiate. Bisporangiate flower is there, kidney shaped. It is kidney shaped, right? Malvaceae family, it is not a tetrasporangiate, it is bisporangiate. It is not a dithecous, it is monothecous. And this Malvaceae family is because we know it is in our syllabus. Already we read in our 11th portion morphology of flowering plants, right? Okay, now here come to the NCERT. A typical angiosperm anther is bilobe. Typical angiosperm anther means mostly they are found, that is called typical. Typical angiosperm anther is bilobe with each lobe having two theca. Two theca means what? Called as dithecus, right? Bilobed dithecus. Often a longitudinal groove runs lengthwise, separating the theca. Right? It is not only longitudinal. That means denses of pollen grain only through longitudinal. Sometimes what happens? A transverse incision is also present. Let us understand various types of tissues and their organization in the transverse section. TSO in the transverse section of anther. Right? What is the bilobed nature of an anther is very distinct in transverse section. Right? Very clear of anther. The anther is a four-sided, that is called tetragonal structure. Right? Structure, it consists of four microsporangia. How many sporangias are there? Four. Because they contain microspores, that means they are called as microsporangia. Which contains spores, they are called as sporangia. Microspore, microsporangia. Megaspore, megasporangia. Right? How many sporangias are there? Four. That's why they are called as tetrasporangiate, located at corners, two in each lobe. Do you understand why they are called as bilobed? These two lobes are there, left and right, tetragonal, right? And each lobe consists two theca. That's why it is called dithecus, right? Two theca means dithecus. Another, it has how many sporangias are there? Four microsporangia are there. That's why it is called tetrasporangia. Tetra means what? Four. The microsporangia develop further and becomes a pollen sac. Microsporangia, when developed, it becomes a pollen sac because a sac in which pollen grains are enclosed. They extend longitudinally all through the length of an anther and are packed with pollen grain. You could see here. This is one sporangia. Two, this is the third sporangia, and this one is the fourth sporangia. Longly, and in this is the basket-like structure, sac-like structure, in which pollen grains are located. Right? Now, another point is our structure of macrosporangium. When we cut this pollen grain in transverse section, what happens transversely? That is called transverse section. What happens? We see the different four layers, right? And in between this, that is in the middle, what is there? Sporogenous tissues. If it is immature, means we can see four layers. But if it is mature, that is anther, then we see only two layers, right? Okay, here, what is given? In a transverse section, a typical microsporangium appears. How does it appear? It appears near circular in outline. Circular outline, right? Microsporangium is circular in outline. It is generally surrounded by four walls. How many wall layers are there? Four wall, and this wall is from outermost. It is epidermis, endothecium, middle layer, and innermost is our tapetum. These four layers are there: epidermis, endothecium, middle layer, and tapetum. Right? Now here the outer three wall layer perform the function of protection. What is the function? Protection, and help in dehiscence of the anther to release from the pollen grain. Here two function is given. Already we will discuss about the separately what are their functions, right? Protection and dehiscence of anther. The innermost wall layer is tapetum. It nourishes. What is the function of nourishment? Tapetum. It nourishes the developing pollen grain. Right? Cell of the tapetum possess dense cytoplasm and generally have more than one nucleus. Means it shows polyploidy or more than one nucleus. More than one nucleus means we can say sinusotic, right? Multinucleated structure. And if it is it shows polyploidy, here amitosis of also there. Due to the amitosis, what happens here? They shows us polyploidy structure, right? And when we consider many nucleus are there, only karyokinesis is there, no cytokinesis is there, right? Can you think of how tapetal cells could become binucleated? Because here cytokinesis is not there, only karyokinesis occurs repeatedly. That's why in each cell more than one nucleus is present, right? If single nucleus, it has a more that is why I say here polyploidy is also there, because we know when a cell is start to divide, what happens, the number of chromosomes, it is okay, they are same, but number of chromatids becomes 4n, right? But sometimes what happens, chromosomes they that means separates, after splitting they separate, but the no new karyogamy or no cytogamy occurs. Then what happens, it becomes polyploidy stage, right? Polyploidy stage means it is called as a mitosis. When the anther is young, okay, a group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporogenum, right? Okay, when it is young, so we have these four layers we can see in a young anther, and also in middle a sporogenous tissue is formed, right? So first we are going to see the structure. I think it is given in our NCERT. A structure is given here, right? Now here look the structure, especially then we will discuss. First see this structure. It is the total stamen structure, right? What is given in this structure? This is the outermost. Outermost means it is given here epidermis. Okay, this given epidermis. This one is epidermis, right? It is single layered. Single layered. Epidermis is single layered, right? This one is connective tissue. Is connective. Connective means what? It is the function. It contains vascular bundles, means water and food, right? Because it connects with filaments. Here filament is connected. Filament is attached through this one, right? So through filament what comes here? Food from leaves and water, right? Now this is the second layer. This second is called endothecium, which is given endothecium. What is the function of it? When you see the endothecium, right? Look here. This is our epidermis. Epidermis, single layer out. Then endothecium. Endothecium cells are the large cells, right? And in endothecium, this endothecium is responsible for dehiscence, pollen dehiscence. Pollen dehiscence. It is responsible. How it is responsible? Because this endothecium, it's radial, and this one, these walls, right? They are thick. Here they are thick in structure, right? But look this side, this is not, right? It is thin, right? These are radial walls, radial wall, and this basal wall, right? In this basal wall, they consist alpha callose bands. Alpha callose. What is the function of alpha callose band? Actually, this is carbohydrate, means it is a sugar, right? And it is hygroscopic in nature. Hygroscopic in nature. Because they are hygroscopic means what? They absorb moisture from atmosphere, right? When they absorb moisture from atmosphere, what happens? They are normal. But when nature water scarcity is there, then what happens? Without water, what happens? They made a pressure on which one? On the wall of look here. In some wall, in some places, what happened? Like this one, right? in this place what happens? It is called stomium. S-T-O-M-I-U-M. Stomium means here thickness is absent. These are thin wall, right? Stomium means this alpha callose band is absent. In stomium, this is stomium. Stomium means alpha callose bands are absent. In some places, in some cells, in endothecium, some cells of endothecium, they lack alpha callose bands. And these places are called stomium, and this is the site from where the anther dehisces, right? So they create a pressure on this stomium, and what happens? These cells rupture because they are thin wall, so they rupture. When they rupture, means now they form, that means they forms a pore or a groove, or we can say that is a slit-like opening is there from where the anthers are dehisced. So this endothecium is the major part which is responsible for pollen grain dehiscence. Do you understand this word? And their alpha callose band is responsible. This is the second, endothecium. Endothecium is also single layer. This endothecium, it is also single layer. Means one cell layer, right? Third is our middle layer. This is our middle layer. It is given here, this, right? Middle layer. Middle layer means it is 1, 2, 3 layered cells are there, right? Either 1, 2 or 3 layers as well. And it is made up of parenchymatous cells. They are made up of parenchymatous cells. So what is the function of parenchymatous cells? They are responsible for storage of food. Food storage. So which one? Middle layer, which is 1, 2, 3 layers. Look here. These are middle layers, right? These middle layer. 1, 2, right? Middle layer. It is here 2 layered structure is given here, right? What is the function of this one? Food storage, right? This steptomal, food storing 2 to 3 layer, and the innermost, this is the fourth layer. Look, epidermis, endothecium, middle layer, and this one, in which dense protoplasm is there. It is called tapetum. Innermost layer is called tapetum. This is tapetum, right? What happens tapetum? Tapetum, here only single nucleus is given, but we know sometimes they have 2 nucleus or more than 2 nucleus, or they have also polyploidy stage due to the mitosis, right? Here what happens? Look, they get food or nourishment. These tapetum cells, they get food from which one? Middle layer, right? So they consume food from middle layer and transport this food to sporogenous tissues, right? That's why in young anther, all these 4 layers are present. But when we that is get a transfer section of mature, here what happens? This middle layer is absent, right? Because middle layer means this tapetum, it consumes all the food materials from the parenchymatous cells, and so what happens? These middle layer, these cells, they get degenerated, right? These tapetum cells, they get food from parenchymatous cells, right? And then they transport this food to sporogenous tissues or spore mother cells or pollen grain, we can say. Tapetum are very important because tapetum is, it is not only responsible for nourishment of pollen grains, but it also secretes some enzymes, right? Like as callase enzyme. Which one? Callase enzyme. And we know callase enzyme because when a pollen tetrad is formed, what happens? It is tetrad means it is a combined structure, means all the pollen grain, they are attached, 4 cells are there, and these 4 cells, they are attached through callose bands, right? So tapetum, it secretes callase enzyme, and this callase enzyme, it dissolves the callose sugar, and then all these, that is 4 cells, daughter cells, they get separate and develop into a pollen grain. First, they secrete some. First, what is the function of tapetum? Nourish the sporogenous tissue. Second function of tapetum, they secrete some enzyme which is responsible for, that is meiosis, or for, that is like callose, right? for break down the callose bands. Third, they also secrete some hormones for developing for embryo, that is pollen grains, male gametophyte. They also secretes Ubish bodies. Look here, these steptem cells, actually what happens here, some granules, you can see they are granules, right? So these granules, actually they are pro-Ubish bodies, right? Pro-Ubish bodies, so, right? These pro-Ubish bodies, they convert into Ubish bodies, right? The pro-Ubish bodies, when does they convert into Ubish bodies? When these pro-Ubish bodies, they contain sporopollenin, right, which is the product in this steptum, right, then they are called as Ubish bodies. Now these Ubish bodies, when these steptum cells degenerate, finally they degenerate, then what happens? These Ubish bodies, they are released into the sporogenous tissue in which pollen grains are formed, right, and this sporopollenin, which is also secreted by our steptum cells, they form the exine, means outermost layer of pollen grains, right. So production, that is production of sporopollenin, production of Ubish bodies, hormones, enzymes, and also nourishment of sporogenous tissue. These are the function of steptum, right. That's why steptum is very important, and it contains a dense protoplasm, and also it has more than one nucleus, right, because it has more ones. Okay, there is these are sporogenous tissue. Each sporogenous tissue cells is each sporogenous cells means in the sporogenous tissue, each cell, it performs like a spore mother cell, and that means what? It acts as spore mother cell. Through meiosis, each cell forms 4 microspores. They perform a microspore mother cell, or sometimes we said them pollen mother cell, right? After meiosis, they produce microspores or pollen grain, right? So do you understand? Single layered epidermis, single layered endothecium, which is responsible for dehiscence of anther. 1 to 3 layered middle layer, which is for storing food. Then single layered tapetum is there. It is multinucleated, binucleated, or polyploidy condition is there. Dense cytoplasm is there because it has many functions. I already tell you this. We already discussed about this function of tapetum, right? And in between it is sporogenous tissue in which each cell, right, act as microspore mother cell or pollen mother cell. After meiosis, they forms the pollen grain, right? This is a mature pollen grain, right? Then after, now here you understand, this is not a epidermis, but these are the endothecium. And here what happens? Here, because here thin walled cell are there, that is called stomium. Stomiums are present that side. Stomiums where alpha callose bands are absent, and here alpha callose band. This is the alpha callose band which you can see just below the epidermis. Okay, and these are pollen grains. These are pollen grains which formed after the meiosis of pollen mother cells or microspore mother cells. As each cell of sporogenous tissue is capable of give rise to a microspore tetrad. Tetrad means 4, cluster of 4. So that is tetrad. Why it is called tetrad? Because here cluster of 4 microspores are there. So they are called as microspore tetrad, and they are interjoined with each other through callose bands. Each one is a potential pollen grain or microspore mother cell, right? Which one? Each one means each sporogenous cell, right? Not, we cannot say each microspore. Each one means each sporogenous cell of sporogenous tissue is a potential pollen or microspore mother cell. The process of formation of microspore from a pollen mother cell, that is pollen mother cell, through meiosis is called microsporogenesis. Microsporogenesis means formation of pollen grain, right? The microspores, as they are formed, are arranged in cluster of 4 cells. Arrangement is also different type of arrangements are there. They may be tetrahedral, they may be linear, they may be T-shaped, right? They may be isobilateral. Isobilateral is very common in monocots. tetrahedral, it is very common in our dicots, right? So they are cluster of 4 cells, the microspore tetrads. As the anther mature and dehydrated, then what happens? The microspores dissociate from each other, right? How they dissociate? Because callus band, we know callus band, it is hygroscopic in nature, and when dehydration occurs, means no water is there, then what happens? It becomes shrink and shrink. And one thing is another, because when these cells, stepped up cells, they get destroyed, what happens? They release Ubish bodies. Also, they release callase enzyme. So, callase enzymes, it dissolves the callus bands, first thing, and this Ubish bodies, they secrete sporopollenin, and this sporopollenin forms a exine, right, around the each cell of tetrad. This forms a microspore, means pollen grain, right? And the microspore dissociate from each other and develop into a pollen grain. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of the anther. Figure is also given. So, first I am going to tell you about the formation of microspore. Here we can say microsporogenesis. What happens in microsporogenesis? That means what? Formation of pollen grain. Pollen grain are also called as male gametophytes. That is male gametophyte, right? First, what happens? Any cell, that is any cell of each corner of corner of A, that is, we can say it is, because it is not a differentiated part, right? A, which type oval shape of oval shaped, it is very young pollen grain, right? Pollen grain, not pollen grain, it is anther, right? Oval shaped of anther primordia, right? Any cell of each corner of oval shaped, it is oval shaped of anther primordia, right? Which one? Which is just below the epidermis. Which we can say it is which is hypodermal. That means just below the epidermis. It stores food and get enlarged. Stores food and get swell or enlarge. This cell, this is first cell. This cell is called, this cell is called, what is called? It is called as archisporial cell. Archisporial, S-P-O-R-I-A-L, sporial cells, right? Now, this archisporial cells, it is the first cell. We can say it is the primordial cell or archisporial cell. This archisporial cell, right? Not it is A. A means what? Archisporial cell. It divide transversely and form forms how many cells? 2 cells are formed. 2 cells, right? Outer means toward the epidermis. This outer which is toward the epidermis, it is called parietal cell, and inner means what? Toward the central region, right? Outer means what? Toward the epidermis, right? And inner means what? Toward the central region. And inner is sporogenous cell. Sporogenous cell. Two cells are formed. Outer parietal cell and inner is sporogenous cell, right? This parietal cell, right? This one is sporogenous cell. Now parietal cell divides anticlinally and as well as periclinally to form 3 to 5 layer of coverings. 3 to 5 layer of covering. Now here what you understand, outermost is epidermis. The epidermis is already formed, right? Second, endothecium. This parietal cells, they are responsible for making endothecium, middle layer and tapetum. You understand? They form this. That means they forms endothecium, second middle layer and tapetum, right? They form these structures. Now the second means what? This is our sporogenous tissue. Sporogenous cell. It divides mitotically, right? It divides mitotically, through mitosis, and forms a homogeneous means same type of cells are there, homogeneous tissue, that is sporogenous tissue. Right? And all the cell of this sporogenous tissue, or we can say also each cell of this sporogenous tissue act as microspore mother cell, or we can say pollen grain mother cell, because they have diploid in nature, because division is mitotic, right? Then what happens? Each cell, each microspore mother cell, or we can say pollen mother cell, divides through meiosis. Now meiosis is there, and then that means they form 4 haploid daughter cells. And that is pollen grain, which are means arranged in the cluster, hence called as, that means they are called as tetrad, microspore tetrad, TETRAD, right? Now they may be, arrangement means what? Arrangement is different type of arrangement is there. Now you can understand here, right? Like this one, they may be This one is called tetrahedral arrangement. It is most common in dicots. Most common, right? Second, isobilateral. It's called isobilateral, most common in monocot, right? This one is linear. This is called linear, right? Decussate. Another one is our it is called decussate. Decussate. Another is here it is called T-shaped. So different type of pollen grains are present, right? And one important thing is there, most common is in dicot is tetrahedral and most common in monocot is isobilateral. And one plant is also there in which all these types of arrangement is present, and this present arrangement is like as below. Below I draw this one, right? And all these types of arrangement is present in a single plant, and this plant example is that is called as Aristolochia. It is a plant in which all type of tetrad is present. All type of tetrad is present, right? Aristolochia. Now these are the tetrads. There in between, what is there? There it is. These are called. This one is called callose band. Callose band means they are interconnected with callose bands, right? That's why when that means tetrad is formed, then what happens? These anther get mature, and at that time tapetum has no food materials. They get destroyed. When they destroy, they release Ubish bodies inside the Ubish bodies, and this enzyme, callase enzyme inside the microsporogensis tissue. This callase enzyme, it digests these callose bands, and then what happens? All these 4 tetrads, all these 4 microspores, they get free. When they get free, because this tapetum cell, they release Ubish bodies. In Ubish bodies means they are sporopollenin. So these each cell of the tetrad, that is each microspore cell, they covered by the sporopollenin, and this sporopollenin, it forms the exine, right? And when a microspore is covered with exine in time, now it is called a pollen grain, right? One thing, okay, we will read this one in. Pollen grain. I think you understand what is how a pollen grain is formed. Pollen grain. The pollen grain represents the male gametophyte. Pollen grain, it represents the male gametophyte. If you touch a open anther, of hibiscus. What is hibiscus, right? Male gametophyte means what? Pollen grain. Or any other flower like Brassicaceae, another flower, you will find disposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on the glass slide, and we can observe in a slide we get this type of structures, right? Because these are ultra structures. not as same as to these structures, but we can, it is the, what is meant scanning electron micrographs. It is electron micrograph, but we can see like these structures, right? Very small grains are, we can see. You will really be amazed at the variety of architectures. Variety of architectures means, look here, different types of exines are there, right? Size, shape, color, designs seen on the pollen grain from the different species. Means different species have different type of size of pollen grain, different type of shape of pollen grain, different type of color is pollen grain, right? Designs is also in that. Pollen grains are generally spherical and measuring, what is the measure? It is 25 to 50 micrometer in. Micrometers means what? 10 power minus 6 meter, right? In diameter, it has a prominent two-layered 25 to 50 micrometer in diameter, spherical, right? Prominent two-layered wall. Outermost is exine, hard outer layer is called exine, which is made up of sporopollen. Made up of sporopollen, which is one of this sporopollen is the hardest part in plant kingdom, and in animal teeth is the hardest part, right? Which is one of the most resistant organic material known. First thing, it is non-biodegradable, right? It can withstand high temperature and strong acids and alkali. One very amazing thing is there, no enzyme that degrades sporopollen is so far known. We have no enzyme in which we can degrade or decompose this sporopollen. That's why in fossil we get pollen grain in good condition. Pollen grain exine has prominent apertures, that is called germ pore, right? Pollen grain apertures, they are called a germ pore, where sporopollen is absent. Means sporopollen is not a continuous layer on the pollen grain. Now here look, sometime what happens, in some cases what happens, this is the pollen grain, right? And this is exine. Here only, here it is absent, it is called germ pore. Germ pore, right? Where it is absent. Look, intine is continuous there, but exine is not continuous. Here only one germ pore, if only one germ pore is there, so it is called as unicolpate. Unicolpate, single germ pore. If germ pore is more than one, look here, one germ pore, second germ pore, third germ pore, 1, 2, 3 germ pores are there. If germ pores are 3, then it is called tricarpellate. Tricolpate, right? Tricolpate is most common in dicots, and unicolpate most common in monocot plants, right? So each pollen grain, it has only a single, or it may be more than one germ pores are there. Okay. Pollen grains are well preserved as fossil because of the presence of sporopollen. Why they are well preserved in fossil? Because of sporopollen, and sporopollen, it is non-biodegradable. That's why it is the well preserved, right? The exine exhibits the fascinating array of pattern and design, different type of design, different type of patterns is there in different type of different pollen grains, right? What do you think? The exine should be hard. What is the function of germ pore? What is the function of germ pore? From germ pore, pollen tube arise, right? Pollen tube comes out from this germ pore, right? Why exine it should be hard? To that is, it preserve the genetic materials, right, in hard and conditions also. The inner wall of the pollen grain is, now second wall, it is intine. It is called as intine. Here a figure is given here, right? Outer exine, inner intine. You can see this one, right? This is a pollen grain. Intine, exine, intine. But here germ core is not given, right? This is a general, we can say it is a plasma membrane. So in the intine, it is a plasma membrane structure. Actually, vacuoles are present, nucleus is present, asymmetrical spindle shape. Means what happened? Here division, cell division, nuclear division is occurs. Now in nuclear division, what happens? Two types of nucleus is formed. One is large, another one is small. One is peripheral, and one is central. Right? Now when they surround by the cytoplasm, they form also two cells. That is vegetative cell, which is very large and central, and a peripheral, that is generative cell, which is very small. After some time, what happens? This generative cell, it enters into the vegetative cell, right? It becomes a spindle shape and enters into the vegetative cells, right? So this is two-celled stage of pollen grain, which is called mature pollen grain. So two-celled stage of pollen grain is mature pollen grain. Why it is considered mature pollen grain? Because at two-celled stage, more than 60% of angiosperm, in more than 60% of angiosperm, pollen grains are shed off at two-celled stage. In 40% means they shed at three stages, right? One thing is very important here. Pollen grains are mature, or we can say a mature pollen grain has two-celled stage, right? Because at this time they shed. But a mature male gamete, it has three-celled stage, right? A mature pollen grain is two-celled, but a mature male gamete, it is three-celled stage, right? It is a thin and continuous layer made up of cellulose, what is intine, inner wall, thin and continuous layer. Exine is discontinuous layer. It is a continuous layer which is made up of cellulose and pectin, and that's why we can say also pectocellulose, right? Pectocellulose. The cytoplasm of pollen grain is surrounded by a plasma membrane. Outer wall is pectocellulose, then a plasma membrane. When the pollen grain is mature, it contains two cells. A mature pollen grain, what is given? Mature pollen grain, right? It contains two cells. One is vegetative cell, another one is generative cell, right? The vegetative cell is bigger and has abundant food reserves, but and it is a large irregular shaped nucleus also. Nucleus of vegetative cell is irregular. Okay, here it is not a circular but irregular shape, right? And the generative cell, it is small and floats inside the cytoplasm of vegetative cell. Initially, it is at one side, peripheral cell is formed, but after some time, what happens? It becomes a spindle cell and enters into the cytoplasm of the vegetative cell. When it enters into the cytoplasm of vegetative cell, now this pollen grain get matures, right? And it is shed off. One question I think in your mind is given: why this cell get embedded inside the vegetative cell? To get nourishment, right? It is a spindle. Now here, generative cell is small and float inside the cytoplasm of the vegetative cell. It is spindle shape. Spindle shape means what? This shape. Cell, right? Spindle shape with dense cytoplasm and a nucleus. First, it is single nucleus. It has single nucleus, right? Over 60% of angiosperm pollen grains are shed at this two-celled stage. That's why they are called as mature pollen grain, right? Because maturity means what? When they are shed, when they become free, right? In the remaining species, remaining means 40%. 40% species, the generative cell divides mitotically, because already when spore tetrad is formed, meiosis occurs, right? In gamete formation, what happens? Suppose that it is a plant, the plant is sporophyte. Sporophyte means it is diploid. What happens? For the formation of gamete, a meiotic division occurs. Meiosis is there, right? But when the plant body is haploid, as in lower plants and bryophytes, for the formation of gametes, no mitosis, no meiosis is there, right? Only mitosis is there. So already meiosis occurs in the formation of tetrads. Now only mitosis is there, right? Because already one meiosis occurs, right? Meiosis occurs only once in life cycle. That is right. What happened? The genitive cell divide mitotically to give rise to male gamete before pollen grain are shed, and this is called three-celled stage. So do you understand here? 60% of angiosperm deer pollen grain shed at two-celled stage, and 40% of angiosperm deer pollen grain are shed at three-celled stage. That is important, right? And in mature pollen grain means, when does it call mature pollen grain? A two-celled stage, a two-celled pollen grain is called as mature pollen grain, but not a mature male gametophyte. Mature male gametophyte means when all the cells are found. All the cells means when it becomes three-celled stage. Then it is called mature male gamete. Okay. Pollen grain of many species causes several allergies. Now, what are the characteristics, that is some properties of pollen grains? Pollen grains of many species causes severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disease. What is given? Chronic respiratory. They are allergies. Some. As for example, which parthenium, that is called carrot grass, right? So their pollen grain is responsible for allergy, right? Allergies and bronchial affliction means through inhale we get this pollen grain spread over lungs. Then what happens? Asthma, bronchitis, these orders may be formed. Chronic respiratory disease. Chronic means what? Long term, right? Because we have two terms, acute and chronic. Acute means what? Short term diseases, and chronic disease means what? Long term diseases, right? It may be mentioned that parthenium or carrot grass, this carrot grass is not origin in our India, because it comes, right, through the imported wheats from US, right? That come to India as a contaminant with imported wheat, and this wheat come from USA, United States of America. From there, when we import the wheat, right, at that time this carrot grass come, and this time it is separate very fast. Means we can say maybe quite a in nature it becomes. Pollen grains are rich in nutrients. Some pollen grains, so they have rich in nutrients. That's why it has become a fashion in recent years to use pollen tablets as food supplement. In Western countries, a large number of pollen grains or pollen products in the form of tablets and syrups are available in market, right? Because they increase our performance, right? Also, pollen consumption has been claimed to increase the performance of athletes and race horses, because they have creatine, and this that means this nutrient, it increase the formation of ATP, and that's why in athletes they increase their performance by 10%. 10% they increase their performance, right? And that's why they are using, some figure is given here, you can see this one. When once they are shed, pollen grains have to land on the stigma before they lose viability. Viability means when they are shed, after shedding, right, they are active, the period in which they are active to that means germinate a pollen grain, right? That is called viability. Before they lose viability, if they have to bring about fertilization, right, how long do you think the pollen grain retain viability? It is different in different plants. That is means variability is also there. The period for which pollen grain remain viable is highly variable, and to some extent depends on the prevailing temperature, and temperature is 25 to 30 degrees centigrade, right? Sometimes it is given 20 to 30 degrees centigrade, and humidity. These two things are responsible for viability, right? In some cereals such as rice and wheat means Grammy family, right? The pollen grain lost viability within 30 minutes, only half an hour, right? If they reach to the stigma, they germinate the pollen grain. Otherwise, they lost their viability, means they destroy. But in some members of like Rosaceae Leguminaceae and Solanaceae, right? What happens? They maintain their viability up to months. You may heard of storing semen sperms in many animals, including humans, for artificial insemination. It is also possible to store pollen grains in pollen banks, right? For many years, of a large number of seeds for many years, in liquid nitrogen, and its temperature is, minus 190 degrees centigrade. We set them the sperm soon, right? Minus 190 degrees centigrade, ultimate 0 degrees centigrade. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding and crop breeding programs. Okay, today we read the microsporogensis and the structure of stamens, right? Next chapter we will discuss about the second one is about the megasporangium, pistil and megasporogenesis. Thanks.